This method can help answer key questions in the plant molecular and cell biology field, such as how can we visualize proteins interacting in the cell. The main advantage of this technique is the high efficiency of co-expressing several fusion proteins in one expression vector. Demonstrating the procedure will be Guitao Zhong, a graduate student from our lab.
To begin, design the primers for molecular cloning of DNA fragments as described in the text protocol. Amplify DNA fragments that are necessary for the construction of the semi-independent protein expression cassettes by standard PCR reactions with their corresponding primers and high-fidelity polymerase. These include the promoter, fluorescent reporter, target gene, and terminator. Examine the quality of the first-round PCR products by looking for DNA degradation and contamination with DNA electrophoresis using a 1% agarose gel. Quantify the PCR products with a spectrophotometer. The ratio between the readings at 260 and 280 nanometers of the PCR products should be between 1.6 and 1.8. Mix the DNA fragments designed for the same protein expression cassette together in one PCR tube to a final volume of 5 microliters.
Avoid mixing DNAs from different expression cassettes together, since it will reduce the efficiency of DNA assembly due to the increasing numbers of DNA molecules that need to be linked.
Now, add 15 microliters of 2x master mixture to the 5 microliter DNA mixture and incubate at 50 degrees.